Our third speaker today has a huge experience in data mining, estimation, and predicting models for scientific domains, with an exclusive focus on computational biology, medical, and healthcare informatics. He finished his undergraduate in civil engineering at IIT Roorkee back in 2002, followed by an MS in civil engineering at the University of Minnesota, but later went on to do a PhD in computer science and engineering in the same university. So he is a distinguished name in the field of data mining for computational biology, and he's currently working for Strand Life Sciences, pursuing R&D in computational biology, clinical applications of genomics. So I welcome Dr. Rohit Gupta.
Hello everyone, I am Rohit Gupta, and thank you for the kind introduction. So I believe there are 3 talks today on computational biology. So this talk is not going to be very mathematical, it's going to be an applied talk. And what I'm going to do in the next 40-45 minutes is to introduce you to some problems in genomics, particularly talking about how can we measure gene expression using a large scale, massively parallel RNA sequencing data. And I believe post-lunch, my colleague is going to talk about some mass spectrometry data. And there is another talk by Dr. Binay Panda in the afternoon. I believe that talk is going to be more general about computational biology and informatics. So I think my understanding is that this particular set of audience, this gene expression and RNA sequencing data is possibly new. Maybe you're not aware of what these things are. So I'll start with some basics to give you some terminology, and then talk about why should we measure gene expression and how to do that using the sequencing data. So going back to school level biology, just spending 5 minutes to give you the background. So all these multi-cellular organisms start life with a single cell, and cell is the basic fundamental unit of life. This individual single cell has to grow, divide, and then differentiate into different cell types, producing different tissues and organs. And you might remember that this is a cell structure where we do have a nucleus in there, and then there is a cytoplasm. So in the nucleus, you would have some important molecules, which is what we want to focus on for today's talk. So we need to focus on something called DNA, RNA, and proteins. So let us just quickly see what these different elements are. So DNA, as you probably all know, is a helical double-stranded structure, which is shown on the slide, and it is made up of 4 nucleotides, which is A, G, C, and T: adenine, guanine, cytosine, and thymine. And DNA is the basic hereditary unit of human being, and all of our hereditary information, genetic information, is carried in DNA, and this is called the blueprint of human life or any organism's life. Now the second concept here is RNA, which transcribes the information that's there in DNA and make real proteins, and proteins are the one that actually do biological processes in the human body. And RNA, as can be seen on the slide, is a single-stranded structure, and pretty much also made up of all these 4 nucleotides, except that thymine is replaced by uracil. Now, proteins are, as shown on the bottom. Protein is the actual unit that derives the biological processes and functions, and proteins are made up by RNA, and they are nothing but the chains of amino acids. And then lipids are there, another form of molecules that stores energy and are responsible for some structural functions as well. So one more slide on some terminology. So all of that DNA information that I just mentioned, every cell, human body has trillions of cells, and every cell has the same amount of DNA. All the DNA that's there, it's there in every cell, exactly same copy. The way that information is captured, the way that information is organized, it's actually organized in the form of chromosomes. So as you might have heard, and it's actually shown here, that you inherit one copy of the human has 23 pairs of chromosome. One chromosome comes from your father and one from your mother, and it combines together to form 23 pairs, 46 chromosomes. So this DNA is actually stored in the form of these chromosomes, as shown. and special parts of those chromosomes are called as genes, which are inherited as from generations to generations. And those genes are the basic units that are responsible to make proteins. So what percentage of this DNA actually is genes? It's only 1 to 2%. So if we have 3 billion base pairs of DNA, human has that much, only 1% or 2% of that is actually encoding for genes and proteins. So only that part is the coding part. And on the picture you are seeing the image of this different 23 pairs of chromosomes. So this is called a karyotype, and this is a karyotype of a normal human being. The complex diseases that we are trying to model and we are trying to understand, like if you're looking for any specific type of cancer, this karyotype is not going to look like this. So there will be large scale changes that you would notice in this karyotype. So some chromosomes are going to be duplicated, some chromosomes are going to be deleted, some portions of the chromosomes are going to be mutated or amplified or deleted. And the objective of this research to some extent is to identify those changes in patients and then connect it back to what are the possible therapies that we can design based on the specific changes that we have observed in the patient. So one dose fits all type of methodology that has been going on in the area of medicine is not going to be applicable anymore going forward. So same drug for same disease is not going to work for all patients. You have to understand what is unique about this specific patient or this specific disease, and then plan therapies according to that. So it's going to be symptoms plus your genomic information combined together. That's going to be useful in going forward, right? So let's begin with some basic questions. So I just gave you information that every cell out of the trillions of cells in human body has the same amount of exactly same DNA. But then the question is, if all cells have the same DNA and same type of DNA, how is variety encoded? I mean, we have muscle cells, we have stomach cells, we have liver cells. All of them are different, right? I mean, the shape is different, their structure is different, their function is different. So what is it that brings variety in these cells? So the question is complicated, but the easy answer is that I want you to know before I get on to the rest of the talk, is that we have all the genes in every cell, but different genes expresses differently in different environments, right? And they differentiate into something different. They make different proteins, and that's why bone cells become hard, skin cells become the way they are, and muscle cells becomes the way they are. So those different signaling inputs make different genes to express in different type of tissues, different type of cells, and they differentiate into something in this much. There's a lot of variety encoded in human body. So then to some extent then it is clear that why should we then study gene expression? Why do we have to understand which genes are active in which situation? First of all, the first application of that is to understand the functions of the genes. We need to understand which cell, which environment has which genes, which genes are active in which environments, and then how to understand the function of those genes, which can be useful to plan for or to design drugs and so on and so forth. The second question is, these days, especially in cancer biology, we need to understand if I do the sequencing of a cancer patient versus a normal patient, I want to understand the tissue is same. Here the tissue is same. Let's say somebody has a liver cancer. I pick up that cancer tissue and I pick up a normal liver tissue from the same patient, and I sequence that tissue. I want to understand, are there genes which are expressed in the tumor sample but which were not expressed in the normal sample, or vice versa. If I understand that, then I can pinpoint, okay, this is the problem, and then the targeted therapies or drugs can be designed accordingly. So that's a very important application, and I'll talk about how to measure those expressions and how to compare the two samples. Now, the third one is again connected, except for cancer, it could be any experimental condition. You can study any organism, you can study yeast, and then understand in different stresses in different environments how the expression of gene changes and what can we learn from that, right? And then of course you can understand at a system level behavior, and rather than understanding individual genes, you can understand overall there are 25,000 to 30,000 genes in human body, how they interact with each other, which ones expresses, which ones are deactivated in different conditions, how they interact, how they make proteins, and how they change the overall behavior that you observe. So one more level of information. So I mentioned in the beginning that DNA is the blueprint. It makes the RNA, we call it messenger RNA, and this is the nucleus. The blue color thing is the nucleus, and this sort of a bread-like structure that you're seeing is the overall cell. So in the nucleus you make the RNA, and then RNA goes out of the nucleus to form different proteins. So in terms of data, the technology is such right now that you can either measure the DNA, you can get the sequence of the overall DNA, or if you want, you can also get the sequence of these RNAs, messenger RNAs. So in the first application, if you are sequencing DNA, I want to understand what are the mutations, what are the changes in that DNA, and are those changes going to affect the protein formation or not. In the second application, if I'm going to measure RNA, I'm going to understand which genes are active in this cell, because if the genes are active, they are going to produce RNA. If the genes are not active, they are not going to produce RNA. So based on the abundance of RNA, I want to understand which genes are active and which are not active. And today I'm not going to cover the first part, just give you a glimpse of that, but cover the second part in a little bit more detail. Right, so here is the gene. There is a start of the gene, there is an end of the gene. It's made up of a lot of things, but only focus on two aspects, exons and introns. Exons are the part that make up protein, introns are not going to do that. That's the only message we want to take today. And here is what happens. Introns get spliced out, exons connect together, and they are the ones that travel outside the nucleus to make the protein. So with that information, let me talk about the mutation part. Why we care so much about the DNA sequence. So if I sequence all of you who are sitting here, we are very similar, right? We are in fact similar to a lot of other organisms. 99 to 98% similarity with chimps and all. You must have read that. And if I'm going to sequence all of you, all of us are going to be very, very similar, except few differences here and there. And that's what makes the unique us. So the phenotypes that you observe, we have different color hair, different color eyes, different nose, different shape of the face, and so on and so forth. We don't really care that much about those differences. Those differences are not harmful, right? But the same changes, some mutations are going to make one susceptible to disease, but the other one is a healthy individual. Those are the changes we want to understand. And it's sort of a needle in a haystack problem. As I mentioned, the genome is 3 billion base pairs. And these differences could be just 1 or 2 or 10 differences in that entire 3 billion code that can make all the changes. So identifying those changes and making sure those are the changes that actually cause the disease is a tough problem, and it's sort of a computational problem, right? So just to give you the third process in that cycle, DNA to RNA, and then from RNA to protein, how RNA is going to make a protein is by using these 3 nucleotide combinations. So rather than reading one nucleotide each, RNA is made up of 4, right? A, G, C, and U instead of T. So we read 3 of them in combination called codons, and each 3 is going to give you 1 amino acid, right? And then protein is nothing but a chain of amino acids. So if there are any mutations, maybe the amino acid is not going to form, and maybe the protein is not going to form, and then the function that protein is responsible for is not going to be carried out, right? But then there is a lot of mechanisms in the human body, natural mechanisms, which protect us from those mutations. So as you can see, if I'm going to read them in triplets, I have 4 nucleotides, and if I'm going to read them in triplets, there are 64 total combinations that I can come across. But we only have 20 amino acids. We don't have 64 amino acids, which means there is redundancy. Lot of codons encode for the same amino acid, and here is the cheat sheet, and you can see that which 3 combinations make which amino acid, right? There are these 20, but I just want you to focus on special 2 types which encode for a start site and stop site. So if you find AUG, that's always the start site, and if you find some other 3 combinations, it's a stop site. Let's see what happens if there is a mutation. So let's say I'm reading them in these triplets. Okay, I told you I'm going to read them in triplets, and these are the amino acids that are going to form. Let's say in somebody's genome, this particular A, which is 1 base out of that 3 billion, it got substituted. A got substituted to G. What would happen if that is the case? Then I'm going to read, instead of reading CGA, the mechanism is going to read CGG, and it may affect the amino acid chain. Let's see from the picture if it actually affects. But as you can see, both are going to encode for the same amino acid. Nothing is going to change. So that mutation is harmless. There is a mutation, but it is going to give you the same protein, and which is driving the biological function. So everything is fine. Now let's pick up another example. In this example, I'm just going to go through this. In this example, the protein is going to change. So I'm not sure. This may or may not have a bearing on the function. The protein may not fold properly. It actually folds into some structure. Because of this change in the amino acid, it may or may not fold properly. If it doesn't fold properly, the function may be affected. So we call this missense mutation. Now, the third is nonsense mutation. Just by the name of it, it's kind of clear that it's even more dangerous, and it is. So what happens here is the chain is stopped prematurely. So because of this mutation, suddenly we have a stop codon in between, and protein stops prematurely, and this is even more dangerous. One more example, we have a frame shift mutation. So instead of substitutions, we may have deletions or insertions in the genome, and because of that deletion, everything changes after that, right? So all of the proteins have changed. So this is another type of mutation that we want to study. So this kind of motivates this. I picked up these examples to just motivate you that why it is necessary to study this sequence. So if we have a technology that can give for each person this whole sequence, and if we have technology to understand what are the changes in this person compared to the reference, then we can understand some of the things, why this person has these deficiencies, why this person is more susceptible to disease, and what can we do, more importantly, what can we do to design therapies. So let's now move on to the data part, right? So now I told you that this is why we want to do sequencing. So sequencing, as you might have heard in some of the, read in magazines and other places, the first sequencing human genome project took almost a decade, almost 10 years. It started in early 90s, and the first human genome was published in 2001, and it took millions and millions of US dollars. Now, nobody could have afforded at that time, and in fact, in the first, in early 2000s, it used to cost $10 million, and even the richest would not be able to afford sequencing their DNA. Now all of us can do that. It's not that expensive. It's almost in the range of $5,000 to $6,000 US dollars. And going forward, there are a lot of research going on to bring the cost down to almost $1,000 US dollars for the whole genome. So it's not that expensive. So cost-wise, the data generation is, I would say, getting really, really cheap now going forward. The challenge still is how to analyze that data. We have huge amount of, you pick up any organism, people have sequenced that organism. Pick up plants, a lot of things have been sequenced. Now, how to analyze that data is a bigger challenge. If I give you your genome sequence, 3 billion base pairs, you have no idea what to do with that. So that bottleneck these days is the bioinformatics part of it, the computational biology part of it, right? So just some technology that no machine can take the entire DNA and read it and give it to you. So we have to divide that into very small segments and then read those segments instead. So you can either read that segment from one end, or you can read that segment from both ends, and that way you get single end data or pair end data. So what are the characteristics of this data? The characteristics are, from a single individual, I get these short reads. The output of this machine is short reads. So out of 3 billion base pairs, the output is only 100 base pairs. So one read is only 100 length out of 3 billion. But I get millions of those. I get millions of those short reads, and I don't know where those reads are coming from, right? So that's the one challenge. Cost is going down, so more and more data is being generated. The cost is actually this. I mean, almost until 2006, 2007, the cost was pretty high, but then it dropped really significantly. So now we are almost here in 2013-14, and the cost, as I mentioned, is almost like 5K for the whole genome, which is not much, right? So the data is huge, but just to give you some sense of what is that huge, and how the genome sizes differ from different organisms, some sort of a fun exercise. So as I told you that human is 3 billion base pairs, and we have about 25,000 genes. Saccharomyces cerevisiae, you must have heard of that, baker's yeast. It's used in making cakes, curd and blah, blah, blah. That's about very small, 6,000 genes and 13 million base pairs. E. coli is, again, a small, 4 million base pairs and 3,000 genes. Right, so can we, do we have a guess on this? How much mouse is, the genome size of mouse in relation to human? Is it close or bigger than human or less than human? Random? Yeah, so it's actually pretty close to human. The reality is the mouse is almost like human. It has 2.5 billion base pairs and about same number of genes. But intuitively, we understand that we are much more complex than mouse, right? We can do much more. That's our intuitive understanding. So we need to understand why. Any guess on this? Salamander, the genome size in relation to human? Larger, that's a good guess. It's actually 30 times larger than human. And it's 90 billion base pairs. Again, one more example, water lily is actually 40 times. The genome size is huge. It's 40 times that of human. It's 120 base pairs, billion base pairs. So one take-home message from here is that the genome size itself, by itself, is not going to determine the complexity of the organism. There is a lot of redundancy. As I mentioned, we have 3 billion, but only 1% of that part is going to encode for genes. Similarly, here there's a lot of redundancy. And the number of biological functions that they can carry out is not going to be linear in terms of their genome size. But the data generation, if you are going to sequence these species, the data is going to be huge. And the challenge of finding that small mutations and connect it to the phenotypes is going to be even larger. Right? So there is a lot of data noise issues, and picking up the relevant information from that size of data is challenging. Okay. So this kind of we covered that why do we need to do sequencing, and what is it that we want to do. We want to find out these mutations and connect it to the phenotypes. A lot of applications in the last 10 years people have found out for the sequencing. You can either completely sequence the human genome, or you can sequence the RNA part, or you can sequence one gene or two genes. A lot of applications that are out there. So I'm going to tell you a few challenges today. Not offer all the solutions, and that is for, I think this is a perfect audience to think of solutions. I'm going to offer some challenges while we face in analyzing such sequencing data. So let's focus on the RNA part today. Why do we have to measure expression of genes, and how can we do that? So let's skip this for now. I think this is a little more technical. We don't need that for today. Here are the computational challenges. If I were to sequence the RNA of an individual, what is it that I have to do with that data? First of all, as I mentioned, I get short reads, 100 base pair, 150 base pair, and I have a long genome. I don't know where those sequences are coming from. So first challenge is aligning them back to the genome to figure out where they actually came from. That's the first challenge, which is, we call it alignment of these short reads. Second challenge is, once we have aligned those short reads, how do we figure out which genes are active and which are not active? Can we just count the number of reads that map to a gene and take that as a proxy for expression of that gene or activity of that gene? Or if not, what are the challenges and how to solve those challenges? That's the second problem. Third problem, which I have not told you so far, I've only told you that there are genes which exist. For each gene, there are multiple isoforms that exist. Can we understand which isoform of that gene is active and which is not active? So that's the third problem. How do we quantify at the isoform level, at the transcript level? The fourth problem, again very important. Now that I have understood or found out the expression of individual genes, can I compare that against two types of tissues, or two types of patients, or two types of populations? Right? So what type of statistical measures do I need? So because if all of us are healthy individuals, and if I estimate the expression of genes, there is going to be some variance in that, right? And let's say there is another group which is a cancer group. You measure the gene expression in that group, again you are going to see some variance. So how do you then figure out that is it the differences that you're seeing, are those normal differences that you would see in a population, or are those differences unique to that disease that you are going to model? So those type of challenges exist, and there are some methods that people have developed to understand or to identify the genes which are differentially expressed across two conditions. And then of course, we have limited understanding. Biologists have been working on different model organisms from ages, but we still have limited understanding of what is a gene. We keep changing every day. And now I'm saying 25,000 genes, maybe 1 year from now it might change. So can we detect novel genes that are unknown right now and figure out what is the function of that gene? So all these are applications.
What do you take as a reference genome or transcriptome, considering that all the sequencing which you do is basically based on a short range?
Right.
So what do you consider as the master?
So that has been done by other group, and they take a lot of data into account. So for human, there is a consortium, the Reference Genome Consortium, and they, after every few years, they try to stitch together different sequences and say that this is a representative sequence for human, and they would say this is the representative sequence for some other organism. So you don't have to design your own reference sequence. It has been a standard that has been accepted worldwide. You use that sequence for species of interest, and then try to find differences of your data from that reference sequence.
But there's still some probability that it's not the perfect.
Absolutely, absolutely. That's why it becomes even more challenging. So first problem is that your data truly is different from the reference genome. Those are the events you want to identify. But reference genome by itself is problematic. There are issues with the reference genome. So all those issues are there, and that's why every couple of years they release new. As and when more data is available, they have more understanding of what the reference should be. They keep releasing new versions of that. But that's a good question. Yeah, that's another challenge that I don't want to talk about today, but it's there. Right, so let's talk about the first problem, alignment problem. I think today we only have time to maybe talk about two problems. One is alignment. We have short reads and we have a 3 billion long string. Just take it up as a mathematical problem. A 3 billion long string data that you have, and you have millions of short strings of 100 size. How do you map it back to that bigger string? So effectively, sequencer is giving you all these reads, short reads, and you have to align these reads to this sequence. And this sequence has 80 GCs, nothing else. And that's what we are made up of, these molecules. So the challenges here is that, first of all, reference is very long, and there are repeat regions. There are a lot of complications in the reference genome. It's not all unique. There are regions which are repeated. So how would you know that this small sequence, 100 sequence, is coming from this area or from that area, right? Now reads, as you pointed out, reads may not have the exact match. I may differ from the reference genome. So I have to allow for mismatches. I have to allow for gaps. I have to allow for all these insertions and deletions. So with that requirement, the mapping becomes even more difficult. So these are the kind of mismatches and gaps that we want to allow. And read sizes could be different. I may have 150, 150, 200. So algorithm should be such that should be able to handle the variety of read lengths, right? And then ideally, I should be able to do it in a reasonable amount of time and memory. I cannot just keep searching for individual reads and spend days and days to do that. So this is, I think, for this particular audience, this is not necessary. I cannot search for these substrings in a brute force fashion. So I have to do something more. So typically what people have done is they have indexed the 3 billion reference genome and use that index to find these seeds, the matches for those seeds very quickly. And once you find the matches quickly, then in that region, you just do local dynamic programming and try to figure out the gaps and mismatches to finally align the read to the reference genome. One of the initial applications were that people figured out there are two types of approaches. In one type, people try to build index on the data, on the millions of reads. In the other type of approaches, people try to index the reference genome and save it. You only have to do it once because the reference genome is not changing every day. You build the index once and then use that index to quickly use your millions of reads to find where they might have come from, and then do dynamic programming in that area to expand for it. So this is an example where you can just, this is a suffix tree that has been built on the reference, which is shown on the top. And if you were to search for a substring, you can actually just quickly do that by tracing the path back. And this pointer leads you to that location where this substring is, right? So you can do that, but the problem is building the complete suffix tree requires, for the human genome particularly, requires a pretty big memory. And as of now, my intention is, of course, people have big computers and clusters, they can use it. But the intention is to be able to analyze these data sets on laptops. And I should be able to figure out a solution which would allow me to run alignment problems of human genome or even bigger size genome on my laptop, which 4 GB or 8 GB memory, and in a reasonable time. So there have been some work in the past, recently as well, where people have tried to compress these structures so that they fit in memory, and that there is some trade-off between accuracy and how you want to compress these structures. But this suffix tree concept was used earlier in few years from now, but it was not picked up really well for this problem. The other type of structure that was picked up really well for the alignment problem is something called Burrows-Wheeler transform. And this has been in use in a lot of other applications, data compression and so on and so forth. But for alignment, a lot of recent algorithms that have been published, they make use of this concept. So you take the entire reference for the human of these 80 GCs, and you try to build a Burrows-Wheeler transform index from that. And it's actually pretty simple. You just write them down in a circular shift manner and sort them based on these letters. And first of all, you have to append some imaginary letter which is assumed to be highest out of AGCTs, and then you just write all the circular shifts and sort them. The last column that you see is called the BWT index, and this column, along with the pointers that you're seeing, is sufficient enough to recreate all these data. So you just save this and try to find the search using that. Now, most of the people have done that, and they do some computations on the fly. But if you were to really, if you have a lot of memory, you don't care about the memory, all you care about the speed, then you want to store something else. So I have told you that you want to store two things. One is the pointers, and second is the last column, which is A and C in this example. But to make the search really efficient, you may want to save two more variables. One is the column B, and column B is very simple. All you have is 4 letters in your DNA sequence, A, G, C, T's. And since you have sorted them out, all the A's are going to be first, and then C's, G's, and T's, and then the $sign. So all you need to save is where the A starts, where the C starts, where the G starts, and where the T starts. So it's just 4 pointers. That's very easy. The other thing that you might want to save from the Burrows-Wheeler transform column, which is on the right side, you may want to capture the rank matrices for each of these letters at every row. So at every row, I want to know how many A's have already been passed, how many C's have already been passed, how many G's and how many T's have already been passed. And that is going to be a killer. I mean, if you were to save all these 4 numbers at every row, 3 billion rows, you want to save a lot of information. But as I said, if my only requirement is time, not the memory, then I want to do that. If my requirement is memory, then I may not want to save all of the D matrix. I would like to compute that on the fly. And that is the advantage of using Burrows-Wheeler transform. It has a very nice property that the whole index may actually take 60 GB of memory. But if I have only 8 GB memory, I can sample all these indexes. I can say I'm going to sample A, I'm going to sample D, so that it fits into my memory. Accuracy wise, I'm not going to be sacrificing anything. It's just that it's going to be taking more time, and it's going to compute few things on the fly. So that's why I think the concept got picked up in computational biology, and a lot of algorithms have been published, and people are still publishing. It's a still active area of research. People are still publishing, especially now that we are trying to go into the clinical domain where lots and lots of patients have been sequenced. So it's no more a research lab problem now. In the hospitals now, people are sending us samples from patients that we do the sequence, figure out what are the mutations, what are the problems in this genome, and how can the doctor make use of the symptoms and this information together to treat patients. So with that volume, the volume has increased. So there are still algorithms being published that how to even, if you have the whole genome or whole exome, how to quickly do it. Trade-off is that you may sacrifice some accuracy because it's not really, you don't really are in the research mode, but the time is more important. So still the algorithms are being published using this Burrows-Wheeler transform. So I'm going to skip that how actually you search the a subsequence, but I think that's there is a blog that we have written. If you are interested, you can actually look into that. And this I already mentioned that although saving everything into the memory requires a lot of memory, but you can always sample it depending on how much memory you have, and that way a lot of other information can be computed on the fly. So this is the blog, and you can just go to blog.avarisngs.com and then you would be able to find this, and there are examples, step-by-step procedure to guide that how Burrows-Wheeler transform is used. That's a nice piece of article there. And as I said earlier, once we have found these exact matches for the short reads, then we can expand using a Smith-Waterman type of algorithms to find to allow for mismatches and gaps, and those would become the final alignments. So this is the approach that we are using right now. I just wanted to give you this problem. If you can think of better solutions from your mathematics background, it'll be very nice, and it'll be a good contribution in the area of genomics. So this is one problem that people are facing these days. This we kind of already covered. Now, the other problem that we are facing, so far I have only told you about the DNA alignment. Let me recap. I told you that gene has two things, exons and introns, and in RNA, introns get spliced out and blue parts, exons come together to form the protein. I also told you that we can directly sequence RNA. So I can directly sequence this part, right, as opposed to the DNA part. Now, what are in addition to the challenges of alignment that I mentioned, what are the additional issues here? So if you focus there, these blue and red part are the encoding parts, and the black line is the intron which get spliced out. So, and then the transcript is going to form, which is what I'm going to read from the machine. So I can get reads which are totally from blue area, I can get reads which are totally from the red area, but I can get junction reads. If I were to use the approach that I just told you, using the genome, that R2 is not going to be aligned anywhere. So that's a problem. How do we handle these junction reads? That's another problem. And then for that, we need to come up with some splice-aware alignment approaches, right? So just wanted to show you that in 90s, people were publishing papers. In 2000s, people are still publishing papers on these things. So it's still an active area of research to some extent, that how do you do RNA alignment? How do you figure out all these splices? One option is you put together all the exons, and rather than aligning the reads to the whole genome, you just align it to the yellow part, which is the exon part. And then you would be able to capture the junction reads, right? And then you put them back where they came from. But this is only good for those organisms where we know what is blue and what is yellow. In a lot of new organisms, we have no idea where the exons are, where the introns are. And if we just take up our existing knowledge of introns and exons, we are going to throw away a lot of data unnecessarily. So that's a problem. We don't have a lot of information about. For humans, we have information. For yeast, we have information. For a lot of plants, for a lot of other species, we don't have that information. So another area of research, another problematic area right now. The second problem, I told you the blue parts, the introns get spliced out and yellow parts come together. The other problem in nature, sometimes the yellow part also is spliced out, and gene has multiple isoforms. So if I have 4 exons in a gene, it can create different types of transcripts, different types of isoforms depending on which exons come together. And identifying which transcript is actually active of the gene is the fourth problem that is open right now. Not open right now, completely open. We have solutions, but another problem that people still worry about. So as I showed you, out of the 3 exons, either all 3 can come together or first and third can come together, and you may have reads from this junction which you will never be able to map if you were to map it to that first yellow combination part. Right, so those are the problems. So let me skip this in the interest of time. Some other problems that I wanted to talk about, but let me then skip the novel part. Let's say we are not interested in finding novel genes and novel exons, but whatever is known, we just want to quantify that. Which gene is active, which isoform is active in different samples. So let's get to that part. So let's say I have done the alignment. Whatever splice aware alignment I had, whatever solutions I had, I have done the alignment of these short reads to the genome. And let's say those dark color things are indicated as genes. So what I can do is I can quickly just count the reads that I was able to map and say that this is the expression of the gene. The more the number of reads I get from a particular gene, that means that gene has more messenger RNA in the sample, and that means that gene is more active. But the problem is not as simple as it seems. So here I'm showing you in the bottom, if you can focus, there is a gene which has 3 isoforms. So all these dark blue things are exons, and depending on which exons come together, you may have 3 different types of isoforms for that gene. So we need to understand together what is the expression of that gene, and then individually which transcript is contributing to that expression. These are the 2 problems that we want to solve now. So and these histograms are nothing but the frequency of the reads that are mapping to that location, and these small gray things are reads, right? So that's the data, and wherever you have more number of reads, you can see the coverage in that gray histogram, right? So there are some noisy reads. Wherever you are seeing this peak, you can see the exon on the bottom transcript. So as I said, we are going to skip the novel part for today's thing. But then there are other issues that one read may map to multiple locations. Then how do you deal with that particular read? Many of the reads can map to 2 different genes or 2 different exons. How do you know where it's actually coming from? So all these type of challenges are there, and a simple type of algorithms that people have figured out is that if there are 2 isoforms, you just create partitions based on the start and end of each exon and count the number of reads for that partition, and then try to figure out which isoforms it actually came from. So that's a simple quantification step that people want to do. For multiple mapping reads, right now what people do is they look at the expression of the gene based on uniquely mapping reads, and then divide the overlapping read in the same proportion, and that has been a standard in this area. The problem is this: ideally, the number of reads that are mapping to the gene should be a proxy for expression level of the gene, because this is the variable of interest to me. If that is the case, I'm all set. But in reality, it's not the case. The number of reads that I'm mapping to the gene, in addition to being dependent on the expression level of that gene, it also depends on the length of the gene. It also depends on what is the sequencing depth of that sample. So let's say one lab has much more money, and they are sampling this individual at a much higher depth. They keep on generating reads from the same RNA element, and the other lab has less money and they don't sequence at that depth. So if the overall number of reads are 10 million versus 50 million, that is going to have bearing on the total number of reads. So you want to normalize for that fact, and that normalization is very easy, right? So the depth of sequencing normalization is easy, length is also easy. So what people typically do to normalize that is something called RPKM. So I'm just going to look at the C, which is my count of reads on the gene, and then divide by the length of the gene, and then divide by the sequencing depth, and use that as a proxy for the expression of the gene. Now, recently people have figured out that this is not sufficient, because what happens, and it's a complicated design. So as a lab PI, let's say I have some amount of money, and I'm going to sample this individual, and I say that I can sequence this individual to have overall 50 million reads. Now this sample has, let's say, many genes which are expressing. So most of those genes would take up the sequencing depth, the share of that 50 million, most of the active genes would take up that, and the other genes would not get enough share. Now, let's say I have some other sample. Again, I'm willing to do 50 million reads on that sample, but that sample doesn't have, let's say it's not a cancer sample or something, it doesn't have a lot of genes which are active. Then majority of the genes would get good share of the sequencing depth. And when I try to compare these two, the genes which got higher share in the second sample might look as more activated in that sample compared to the first one. But the first one, the genes are equally active. They just didn't get enough share because of the design requirements. So it's a problem right now, and it's a difficult area. So that's why people have figured out some other normalization mechanisms to also normalize for something like this. So there are very sophisticated approaches where people are trying to normalize for length and sequencing depth. In addition to that, they try to normalize for the intra-sample variations in the gene. Right? So just to give you a flavor of that. I'm going to talk about the isoform quantification problem that I just mentioned briefly. So in this example, I'm showing you 4 samples. The data is being shown from 4 samples, and all these peaks that you're seeing, they would map to the exons that you're seeing on the bottom panel. So on the bottom panel, I'm showing you one gene, and that gene has 4 different types of isoforms using the exons that are there. And then I have sequenced this thing. The problem is of a deconvolution problem. The signal that I'm seeing is a combination of all the 4 transcripts. I can figure out whether this gene is active or not, but it's difficult to figure out which of these transcripts are contributing to that signal. So it's a deconvolution problem that I want to understand. So just to give you a quick intuition that how it's been done. So we have 2 isoforms, I1, I2. And then I create the partitions that are shown in the orange or this red partitions. And if I tell you that partition 1 has 5 reads, partition 2 has 5 reads, and partition 3 has 5 reads, can you tell me which isoform is active? Actually, the answer is here. So isoform 1 is active, because if the isoform 2 is also active, then I would have seen more number of reads from P1 and P3 and less from P2. But all 3 partitions have same number of reads. That's an indicator that isoform 2 is not even expressed in that particular sample. Only isoform 1 is expressed. If I see 505, then I know isoform 1 is not expressed, 2 is expressed. If I see 10-5-10, that's an indicator of both are being expressed. So that's just a toy example of showing how these things are done. But typically, it's much more complicated. So people try to figure out what are the variables that may affect the quantification or the activity of that specific transcript, and they try to make a model out of that. So here theta is what is the variable that we want to measure. So theta is the activity level of each transcript. Rn is a random variable which is nothing but your data. It's the reads, and let's say you have total n number of reads. And Gn is the isoform, right? So there are 4 isoforms of the gene that I showed you in the previous example. So based on that, they depend on the start position, they depend on the orientation, and this is an older model. This is like 3 years old. There is a recent paper from the same authors, and the model is now much more complicated. They have included more variables. But this is a classical case of that we are measuring Rn, and we want to estimate theta based on some model and some variables that I have in the system. So it's a classical example of, I can just use some sort of EM type of algorithm and try to solve this problem. And right now, what we are doing, that's exactly what we are doing. We write the data log likelihood, the overall log likelihood, and based on the connections that I showed you in the model, I can just marginalize these things. Not every variable is dependent on all others. So I've just done that. And then ultimately, it's a standard EM algorithm. I initialize the theta, and then I say, okay, give me the data likelihood. Based on that data likelihood, I try to re-estimate the parameters and keep iterating. Just use the existing EM algorithm for this problem and understand what is the final transcript levels of each of the isoforms. Now, rather than showing that in equations form, here is the example, where you have 3 transcripts of a gene, red, green, and blue, and you have 5 reads, A, B, C, D, E. That's your data. So from that read and from the transcript information, and here is the mapping. The genome is shown, and the reads are mapped to that genome that has been shown. Based on that, this example, I don't think I have enough time, but and I don't think it's necessary also for the audience like this. Just iteration steps are shown that how expectation step changes the mapping of the reads to these transcripts, and then how M step recomputes the abundance of these transcripts to arrive at something. Ultimately the abundance of transcripts don't change as much, and that you take as the final abundance. So this is what we use to estimate which isoform of the gene is active. Now, I think that's pretty much all that I want to say, because there are other problems, but we don't have time to cover those problems. So I covered two problems today, the mutations in DNA level and how to connect it back to the clinical parameters, and then how do we estimate expression of genes, and how do you find out what are the different genes which are active in one sample versus the other sample, and make use of that information to design personalized therapies. So that's the overall goal of the company. That's where we're heading to. I think that's all I have to say. Thank you for your patience.